Okay, so in this video, we're going to look at mitosis and meiosis. So we'll start with mitosis. Mitosis is the process of cell division for asexual reproduction. Essentially, it involves the replication of DNA followed by the cell splitting in half, creating two genetically identical daughter cells from a single parent. In single-celled organisms, this results in two offspring that are identical to the parent. And in multicellular organisms, it allows for the growth and repair of organs and tissues within the organism. Meiosis, on the other hand, is the process of cell division for sexual reproduction. It involves the division of a germ cell twice, giving rise to 4 cells or gametes, each possessing half the number of chromosomes of the original cell. These gametes then combine with those of another organism to create new, genetically different offspring. So there are some key differences between mitosis and meiosis that are worth noting at this stage. Mitosis is a form of asexual reproduction, while meiosis is part of sexual reproduction. Mitosis occurs in all organisms, but only animals, plants, and fungi can undergo meiosis. In mitosis, the offspring are genetically identical, but in meiosis they are genetically different due to a process called genetic recombination. The process of mitosis involves one cell division, results in 2 daughter cells, each with a complete set of chromosomes, while meiosis involves 2 cell divisions, resulting in 4 daughter cells, each with half the original number of chromosomes. So all the cells in your body, other than your egg or sperm cells, were created via mitosis, and your sex cells, eggs and sperm, were created via meiosis. So what does this look like? Well, here's a breakdown of the process of mitosis. Firstly, during interphase, DNA is duplicated in preparation for cell division. Once the cell initiates cell division, we start at prophase, where the nuclear envelope breaks down and a spindle forms between the 2 centrioles. At the top, you can see a diagrammatical representation of this, and below, you can see an actual microscopic image of a cell at this stage. In prometaphase, the chromosomes attach to the spindle fibers. And in metaphase, the duplicated chromosomes, or chromatids, line up at the metaphase plate, or the equator of the cell. In anaphase, the chromatids are separated and begin moving towards the polar regions of the cell. In telophase, the chromosomes reach the poles, they decondense and nuclear envelopes form around them. The spindle breaks down and the cell begins to divide. And finally, at cytokinesis, the cell divides. In meiosis, you have 3 key modifications to this process. Firstly, the original parent cell divides, and then each daughter cell divides again. This is how you end up with half the number of chromosomes in the final 4 cells as what you had in the original parent cell. Secondly, during the first cell division at prophase 1, homologous chromosomes undergo synapsis or pairing prior to their segregation. This allows for chromosomal crossover, otherwise known as genetic recombination, and this ensures genetic diversity in the daughter cells produced. And thirdly, during metaphase 1, the homologous chromosomes align together at the metaphase plate in the equator of the cell. Remember, they were paired together during prophase 1. So that's basically how mitosis and meiosis work. I hope this has helped you to understand the processes better.